So I set up an email address specifically to do the kit. And it's pretty interesting because there's no return address. So really the only indicator for which tube is mine is this 14-digit code. I spit in the tube, you're smart, you can figure that part out. And you write down that 14-digit code on the tube and link that to the email address. And that goes to 23andMe headquarters. The kit, however, does not go to 23andMe headquarters. It goes to LabCorp, a completely different company. After doing some research, I discovered that 23andMe has isolated themselves physically from the samples. LabCorp has samples and numbers. 23andMe has email addresses and numbers. Sure. What are you guys doing?
I'm currently uncapping specimen samples to be able to put them into the Tecan, which will then take a little small amount of this into the 96 well plate.
Okay, so these machines are autonomously taking the samples out of the specimen tube that people mailed in,
Yes.
and then it's automatically putting those into the 96 well plate. That's what we're doing. Got it. Okay, so this is the first time where we no longer have humans touching it and we're moving towards machine automated process.
Exactly.
Is that it?
That's right.
I would high five you, but you got gloves on. I spent the entire day with Amanda learning the whole process, and basically it works like this. First, they isolate the DNA, and then they amplify it by allowing it to replicate, and then the magic happens. This chip is the secret to genotyping. Basically, they take the DNA and they unzip the double helix, which leaves you with one side of a ladder rung. They then put this unzipped DNA onto the chip, which has thousands of artificial DNA probes on it. These probes are basically long stretches of DNA that match known parts of the human genome, so when a ladder comes along and gets beside the probe, it's a perfect match and will bind to it. Since this stretch of DNA is a known sequence, you can then read the next base pair that's hanging off of the probe, and that's the specific information for that base pair. They do this by a fluorescent marker that they'll put on the chips, which reacts to different kinds of light. These specific locations are chosen because they're associated with something interesting, like a disease risk or a physical trait. This fancy machine illuminates the chips and takes a picture which tells you what letter you have at that particular DNA SNP, or single nucleotide polymorphism. And that's it. That's how the data is captured.